All right, welcome back. Today, actually, wait. Before we do that, before you click off, please like and subscribe the video. My friend told me that instead of saying the end of the video, say in the beginning of the video and see what happens. Do I think something's gonna happen? Probably not, but what? It's worth a try. Today we're gonna be doing neurons and glial cells, which are supporting cells. So let's start with neurons. So a neuron is the functional unit of the nervous system. And in part of neurons is the axon, which carries outgoing information. So let's break this down. What do we see here? So we start always we're going to call this the top. And do you see these little prickly things here on the side, right? Those are called dendrites. And what they do is they receive information from other neurons. Okay? Now, this information is in the form of electrical signals. So and when I mean electrical signals, I literally mean electrical signals. So there was a study done. This person was under anesthesia and they cut open his skull. He was still alive, right? They just opened up his skull and they connected wires to his brain and to a light bulb on the other hand. And he was told to move his arms and whatnot. So when you're under anesthesia, you can be under, asleep, or you can actually be awake, but you don't feel any pain at all. So he didn't feel any pain, and they told him to move his arms and whatnot, and the light bulb actually turned on. So it is, when I say electricity, I actually do mean it's legitimate electricity, not made up. So what happens is the signal goes to the nucleus, which is the red thing, and it gets processed. We're gonna make information. We're gonna make do with the information that's coming in. Then we go over to the axon hillock. What happens is it sums up all the signals that's been received by the dendrites, and it decides whether to send an action potential. An action potential is literally like an electrical, it's basically like a lightning strike or an electrical signal. That's literally what it is. So what happens, so let me draw it out now. So these dendrites receive signal from other neurons. So there could be a neuron here, 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 a neuron here. They're all touching these, dendrites. And what happens is that some of these signals can be positive signals and some of them can be negative signals. What do I mean by that? So we call those EPSP or IPSP, which we're going to go over later. But essentially, it's an electrical signal that tells, all right, I want something to happen, like I want an action potential to fire, or I don't want an action potential to fire. So that, like, the negative signals are called IPSPs, and it's basically saying, no, we do not want an action potential to fire. The EPSPs are positive signals and basically say, yeah, we do want an action potential to fire. We want it to take that message to its destination. We don't want it to stop. Okay, so the axon hillock takes into account all those neurons and all those signals and says, all right, let's see, do you have more EPSPs or IPSPs? If you have more EPSPs, this is roughly how it is, we're going to make an action potential. So it's going to form, like, it's basically all ions, which we're going to talk about, in the next video probably. We get an electrical signal and it goes super fast down this axon, like lightning fast, speed of light, literally. It goes down the axon. Do you see on the axon, there's these blue rectangles. These are called myelin sheaths. They're essentially, um, it's basically, in a way, it's like kind of like a structure, and it protects the axon from signal loss, and it also speeds up the signal. So when you get the action potential, that little, the signal going down the axon, we don't want the axon, the electric signal to leave, right? This neuron or axon, because it'll never reach the destination if you have signal loss. That wouldn't be good. So this myelin sheath protects it. It also speeds up the signal. That there's a mechanism which we're gonna go over later in another video of how it actually speeds it up. And those little gaps you see in here. So the gaps are called node of Ranvier, and this helps speed up the signal. So a fun fact is that if you were to cover this entire axon with myelin sheath and no space, like no bare spaces of axon, it would actually be slow. We would have problems. So we actually need these gaps here called Noether-Van to actually see up the signal. Then we get the axon terminal. This is the end of the line. So think about this as a train, right? We begin at the dendrites, then it goes to the station, which is the cell body, where the nucleus is. Then it goes down the track, and now we're at the destination, we're at the end, the end station, where that is, and that's the axon terminal. The axon terminal has neurotransmitters in basically the tips. Okay, so there's like, let's do here. So there's like neurotransmitters in here. And what's going to happen is when the electrical signal gets there, the neurotransmitters are going to diffuse into this place called the synapse. That's called the synapse. And what's going to happen is these things are going to bind to the dendrites of the next neuron that's next to it. And this would cause, a signal to go here to the nucleus and then back to the axon hillock, and we do it over again. So the first neuron in this stage is called the presynaptic neuron. Presynaptic neuron. We are before the synapse. This is the synapse right here, right? We are before it. So the neuron before the synapse is the presynaptic. This means the one after the synapse is the postsynaptic. So imagine we have another synapse after this, the second neuron. So after this neuron right here, we have another synapse, another neuron. Then this postsynaptic neuron is now called the presynaptic, and the next neuron in the, next up is the postsynaptic. And then this becomes nothing. We don't care about this anymore. Okay? So the whole point of neurons is basically to transmit information in the form of electricity, essentially. And this would cause hormones to be released. This would cause, you actually able to move, able to feel stuff, etc. So many things. Okay. Now we have something called glial cells. These are the supporting cells of the nervous system, and it helps neurons achieve their goal. It sounds like they don't really have much of a role here, but this is actually a lot of stuff. It actually has a big role in our nervous system. So the first thing we're going to talk about is what the central nervous system is, aka the CNS. So any neuron or any glial cells that are part of only the brain and spinal cord in your body is part of the CNS. That's the CNS, only brain and spinal cord stuff. There are 4 microglia present. Sorry, 4. Did I just write microglia? No. There is, I apologize, glial. There's a reason I put microglia. It's because there are microglia cells, which are glial cells. The peripheral nervous system, or the PNS, it's basically everywhere besides the brain and spinal cord. So there's 2 glial cells present in the PNS, 2 different types, I should say. So 4 basically different types. Okay. So CNS is brain and spinal cord, peripheral nervous system, or PNS is everywhere else besides the brain and spinal cord. So the first one we have, the first supporting cell, is called astrocytes. This literally translates to star cell. The reason is because I drew this. They basically look like stars. So all the pictures you're gonna see, I hand drew, because I wanna, make the best quality content for you guys. So what do they do? So they are the source of neural stem cells. Okay, I wanna tell you a fact, and it's very, very important you understand this. So what I mean by stem cells is this is when in development, when you're, inside the mom, whatever, and you're growing. Okay, the neurons are essentially growing. They're forming to become neurons. When you're an adult, they don't do this anymore. This is why it's super, super important that, you protect your body from all causes of harm. So, for example, people who've been in an accident, I want to prove this to you. People who go into a car accident, for example, and they're paralyzed, there's a reason they stay paralyzed. It's because they don't regenerate. These neurons don't regenerate. They can be repaired up to a certain extent, but they cannot be fully repaired. When I mean repair, I mean like very little minor things. So think when you get into a car accident, a really bad one. Astrocytes cannot completely fix a totaled car. All astrocytes can do is basically fix up the paint in your car, and that's it. It cannot fix a totaled car. So you total your neurons, you're done. This is why it's super important. They also take up neurotransmitters. So those neurotransmitters that have been released, right, in the axon terminal, what we talked about, and they're in the synapse, these astrocytes come and clean them up. So they also repair, like I said. They also form the blood-brain barrier. What is this? So in your brain, what separates the cells in the brain, right? We don't want them to like mingle with each other and, cells just be floating around in the blood, right? The brain needs to be protected. How is the brain all in one piece, right? And not just like a mush of stuff. This is what astrocytes do. They also provide substrates for ATP production. So essentially, they feed the neurons. So neurons need food in the form of ATP. Astrocytes help with that. And astrocytes are only located in the CNS, not the PNS. This is very important. You know this. This is usually this is a very common exam question. Astrocytes are located where? True or false, right? Like true or false, they're located in the PNS. False. Only in the brain and spinal cord astrocytes are located, nowhere else. I want to propose a very interesting topic to you guys. A question. Isn't it strange that we have so many cells in our body that regenerate and regrow, but the only two cells that don't do that are nerve cells, neurons, and heart cells? The only two cells that we really need the heart to live, and we really need our neurons to live. Isn't that funny? But for example, our kidneys, right? They regenerate. Digestive system tissue cells, they regenerate. Your lungs tissues, they regenerate slowly, but they do. Your heart does not. This is why you need to protect your heart. This is why people who have heart attacks, you get a heart attack, and they tell you, oh, you need to really rest and don't do anything strenuous. You damage your heart. The cells in the heart, they were not going to regenerate. You have what you have. And that's why you have to protect your heart as much as possible. This is why eating well is so important. You eat junk, you eat fat food, right? Fatty foods and stuff like that. You're going to damage your heart, and once it's damaged, it's damaged. And don't think you can get like a heart transplant or whatever. It's a long process. You have to see if you deserve it, blah, 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 right? Long process. And you have to be on, antivirals your entire life. This is why it's super important. You protect yourself. Next. Sorry, a little tangent over there, but I'm like explaining things that your professor won't tell you or a book won't tell you. The stuff I just told you right now, you won't hear anywhere in the book. Or even a professor, professor just tells you the content. I want to make sure you guys are ready for the real life. You want to become a doctor, you want to become whatever. All right, next up, you have epidemiocytes. These create the barrier between compartments. Okay, so how do the neurons just not touch other stuff around the area, right? These epidemiocytes protect it. They're also the source of neural stem cells, just like astrocytes, but only in development. The main point, and this is why I bolded it, is produces cerebral spinal fluid. So what CSF does, it provides buoyancy and protection for your brain. We don't want the brain bouncing around, right, and cause brain damage. The CSF protects that. I can prove to you what CSF is. If you're ever an E.M.T, or honestly, I hope this never ever happens, but if you get into some kind of brain damage, for example, and you notice a very clear liquid, it's clear liquid, and it's like a funny texture coming out of your nose or ears, that's CSF. That's something that doctors can tell if you have brain damage. or you've injured something part of your brain. If CSF comes out of your ears. I know it's kind of gruesome, but it's kind of interesting how we notice these things. Next is microglia. This is how I made the mistake earlier. Microglia. Oh, I should say epidermal cells only in the CNS, not in the PNS. Microglia, located in the CNS. They literally what they do is it basically translates to small cell or small glia. Essentially, they're really small. They eat foreign particles via phagocytosis. So it literally eats stuff. These cells eat whatever it finds that's foreign. If it's a bacteria, if it's dust, I don't know, for example, right? Anything that doesn't belong where the neuron is, it'll literally come and eat it up, engulf it, and digest it, and it lives. It's only located in the CNS. So the pink things, right, the pink looking, they kind of look like astrocytes, but they're smaller and less branches, and they stick to the neuron. Okay, do you see how they're stuck on there? Right? They're basically crawling on it. They literally crawl, but they kind of stay, it's weird. But they come and eat stuff up. Right? The last thing, the CNS only, is oligodendrocytes. Oligodendrocytes form that myelin sheath we talked about, right? The protection of the axons. Let's go back. Remember, see this blue stuff, right? That's what oligodendrocytes do. The myelin sheath protects axons from electrical signal loss and speeds up signals. This is what oligodendrocytes do, is that they build this myelin sheath. It's only located in the CNS. So people who have myelin sheath degeneration have difficulty walking. It's because the electrical signal, is taking a long time to get to the legs or the brain or whatever. There's a delay. And that's why it's hard for people to walk, if people who have this disease. Next, you have Schwann cells. Now, this is in the CNS. This does exactly the same thing as oligodendrocytes. There's no difference except it's located in the CNS. That's how dumb this is. They just changed the name. Right? There's a guy named Schwann, I'm assuming, and he found this. And now we have to suffer because now they're named two different things. Same thing, same exact thing, except one's in the CNS and one's in the PNS, right? This is CNS. Oh, I made a mistake. Uh-oh. Hold on. Hold the phone. Where did my little, my pencil go? Oh dear. Did it fall? Where did it go? I'm sorry, this is like unprofessional. Okay, let me just use my finger. Or not. Oh, it's right on top of the book. I'm so sorry for the delay. I should check my work. This is why you should always check your work. PNS. It's only in the PNS, okay? I also made the same mistake here. Oh my goodness. Oh, okay, I'm bad. All right, satellite cells is the last one we're talking about. It's not satellite. There's no R in there. It's satellite cells. And it's only located in the PNS. It's the pink stuff. This creates structural benefits for the cell body. How is the neurons held into place, right? How do they not move, right? How does the cell body, how's it staying still in your tissues or whatever, right? In your body, it's satellite cells. That's what they do. It literally just covers the cell body so it doesn't move. That's it. And that concludes axons and slightly, and glio. I hope you found this informational, and I hope, please like and subscribe. You guys are really supportive. 24 subscribers. I'm very happy, and I'm going to keep doing this for years. So, all right, I'll see you later.